Okay. So one of the most fun things that we do in this course, I think, is actually something we're going to do today. And this is really one of the ways that we synthesize everything we've done to this point, and everything we've done to this point in thinking about genes, the biochemistry of the gene, pedigree information, how it integrates with human history, which is really one of the fun things about genetics, is how important it is for evaluating what goes on in our lives and what's gone on in our ancestors' lives. So I'm going to share with you, it'll take me a moment to turn this slideshow on. Here we go. And then, of course, I have to share it with you. And I have a bit of a personal connection here. Is we get a chance to talk about one of the great figures in history. Certainly, if we think about King Tut or Tutankhamun, uh, it is certainly one of, I don't remember if it's officially one of the seven wonders of the world, but it certainly is to me quite a remarkable part of our human history. Uh, I usually, if we were in class, I would show you the Steve Martin video of his music video of King Tut. Great comedy bit. If you get a chance, should go online and look for Steve Martin's King Tut dance. But it's really quite a serious part of our human history, and there are layers upon layers about King Tut. We can talk about so many things. I was very lucky in my career, in that I did quite a bit of international traveling, and my first trip out of certainly what you might consider developed nations was really to go to the University of Cairo to talk about applications of biotechnology in poultry breeding. So it was more an agriculturally focused visit. But along the way, my hosts, I was there for 3 months, and my hosts were incredibly gracious. And I really got quite a tour of Egypt. And one of the places that we visited and spent a long weekend at was a place in Luxor called the Valley of the Kings. And the Valley of the Kings was where all of the major tombs of the pharaohs in ancient Egypt were placed. And it was really kind of, if you think about it in more mundane terms, it was really kind of a graveyard. It's where all of the pharaohs, and remember, pharaohs were godlike creatures back in the day. And several of the tombs had been identified and found. But in the early 1900s, a guy named Howard Carter, uh, quite honestly, when they say stumbled, and when I say he stumbled, he actually stumbled. They were going and walking around. He and his colleagues were going and walking around in this area of the Kings, in the Valley of the Kings, and there was a sandstorm. And unbeknownst to them, it had moved the sand. And as they were walking along, his foot broke through the ground, and he honestly fell into what became one of the first doorways into uh, the pharaoh's tomb, in this particular pharaoh, into King Tut's tomb. Really remarkable if you think about it. What a stroke of fortune. And it took several years. In fact, it took about 10 years. I apologize, but my chat doesn't work. No problem. I can't do chat and this at the same time on my laptop at home. So again, but really quite remarkable if you think about walking around and sticking your foot, walking on the sidewalk and then going across your grass, a place you've probably been a million times, and all of a sudden your foot breaks through and you find the proceeds of the Brinks bank robbery. I mean, that you find millions of dollars hidden in your backyard, and that's kind of what this was like. It took 10 years to completely uncover the tomb. And in fact, it took 2 years until they found the actual sarcophagus of King Tut. And this image that you're seeing right now is actually the sarcophagus. King Tut's mummy he was mummified when he died, and he died as a young teenager. And after he died, he was mummified, and the mummy was placed inside this solid gold sarcophagus. And this is actually the sarcophagus. I was very lucky to be able to see the sarcophagus when I was in Egypt. This occurred I was there in 1996. And at the time, the sarcophagus was in the Egyptian Museum in downtown Cairo. It's still there, but it's very protected. But at the time, you could actually walk up and touch it. They don't allow that anymore, kind of like the Mona Lisa in the Louvre. It's now protected by bulletproof glass. There's kind of a weird story. Someone tried to several major works of art from the Western world there've been kind of crazy people trying to attack them. Somebody tried to break in to actually tear up the Mona Lisa in the Louvre a number of years ago. And so they have if you go see the Mona Lisa now, the Mona Lisa is behind shatterproof, bulletproof glass. The Pieta, one of Michelangelo's classic sculptures. Somebody during a tour went at it with a hammer and broke pieces of it. And so now you can't walk up to it. You have to see it from behind, again, shatterproof glass. In the Tutankhamun story is actually a little bit different. It turned out that the curator of the Cairo Museum was actually cleaning the sarcophagus because, again, it was open to the public and you could actually go in and, put your hand on it. Nobody'd yell at you. You could walk right next to the sarcophagus, touch it. And he was cleaning it. And he tried to lift it by grabbing onto the beard. You could see the beard-like structure is coming out here. He grabbed it by the beard, and when he lifted it, it broke off. And so the strange thing, I guess the funny thing, it just shows you human nature is the same no matter where you are. He tried to glue it back on with like glue from his desk. And then somebody noticed that it was broken. It has since been repaired and fully restored. But I think they transferred him from the Egyptian Museum to the museum, to the car museum or something like that, the ancient car museum in Cairo, which is actually true story, a little weird. So what then, I've got some pictures here. These are actually from the National Geographic. There we go. This is actually the location. This is a view from the top of the road as you enter into the Valley of the Kings. And this is the view into basically the top of this structure right up here. If you see, there's like a little doorway, and the top of that is actually the part that Carter fell through. So this was entirely covered with sand, and that's where he fell through. And eventually they were able to uncover, and they've built some roads now. You can now enter the tomb and go through this pathway. You walk up these steps and you go into the pathway. You can actually go into King Tut's tomb right now. Really quite a remarkable experience. There is not much there. I have heard that they've been trying to put some artifacts back into it. When I went, it was just an empty, it looked like an empty cave. There was really not much there. These are photographs taken, these are posed photographs taken uh, by Carter, who's the man on the right, pointing to where he fell into King Tut's tomb. These were publicity photos taken sometime in the late 1920s, so it didn't happen in real time. These were taken for the museum purposes. Here they are finding the doorway underneath where he broke through, and of course they broke into the tomb, and you can see Carter again standing there, and Lord knows what kind of damage they did breaking in. They were just trying to get inside. You could see the hieroglyphics on the walls behind them. All the walls were covered and decorated with hieroglyphics. You could see the carvings when I was there, but not the painting because all the pigments had faded. And here's what it kind of looks like when you get to the uh, when you get to the Valley of the Kings. And right here in the middle, you could see the Tutankhamun tomb is set just off to the left center of the blue lettering that says Tutankhamun. And you could see all of the different tombs from all of the different pharaohs, ranging from all of the Ramses to Amenhotep, Seti, Thutmose, all of, and importantly, Akhenaten. Akhenaten, you'll see in a moment, plays an important part in our story. And they're all interconnected by tunnels. And at the time of their discovery, they were really covered by sand because of all the sandstorms. So they had to literally be excavated. All the sand had to be moved in order to find these tombs. Sadly, many of the artifacts, even the mummies themselves, were stolen by grave robbers, especially in the late 20s and through the 30s with the World War, especially in World War One, as the Germans took over northern Africa and invaded. Many of the artifacts that were so important to Egyptian culture, not to mention human culture, were raided and stolen. Many have been returned, but we have no idea how many have been lost to history because either they were destroyed or they were in private collections somewhere and no one knows where they are. But really quite a remarkable history here. Here, of course, 2 years after Carter broke through the roof of the entryway, 2 years of discovery and excavation led him to find the sarcophagus of Tutankhamun. And you can see this here. This was a picture taken in more or less real time after the discovery, and you can see the classic headpiece. And here is where he was entombed. And here's a real world picture of what it looked like. Really quite remarkable. Solid gold encrusted with jewels of inestimable value. Inestimable value. I mean, and it is so incredibly beautiful. By the way, these are not pictures that I took. These are from the Smithsonian. These are from Smithsonian Magazine. Periodically, the headpiece for Tutankhamun comes on a museum tour. I do not know when the next time it's going to be allowed. Things are crazy, not just the pandemic, but politically, the world is in a bit of an uproar, not the least of which is the Mideast. But I highly recommend, if you ever get a chance to visit, definitely go. It often hits Boston, New York, or Philadelphia, so they're within driving range and certainly visiting range. I highly recommend you going to actually see it in person. Even if you can't get up close and touch it anymore, it's just remarkable to see something like this in person. So here we have a photograph of some of the hieroglyphics that were found, and this is actually the process of embalming King Tut. And this is partly servants, partly family, and they are embalming him and getting him ready for burial. Remember that in the ancient religion of the pharaohs, these paintings were meant to communicate to the gods, and many of the valuables were there as offerings to the gods. And of course, one of the things that the gods were to do were to reassemble and revive the body of the pharaoh so that uh, they could enjoy the afterlife. So quite something. And King Tut was very, very young when he died. He became pharaoh when he was either 9 or 10 years old and was an early teenager when he passed away. He was very, very small of stature. It's very often thought that he had some kind of a debilitating bone disease. If you think about the breeding or at least marriage practices, I guess if we were talking about animals, we would talk about breeding practices, but these were cultural practices of the pharaohs because they were supposed to be directly related to the gods. Pharaohs could not marry and or have children with just anyone. They had to have children with other deities, and that way to maintain a pure bloodline. As a result, some of their pedigrees there are a lot of full brother, full sister marriages, uncles and nieces marrying, all sorts of strange combinations of marriages. And as you might imagine, inbreeding increased very rapidly. And so many of them had infant mortality, probably a lot of diseases, many we don't know about because the people passed away. However, we do have the remains of many of the mummies from all of the tombs in the Valley of the Kings. And in that regard, a number of years ago, this actually began on the order of about 20 to 25 years ago, the thought was, as we learned about DNA, can we extract DNA from the mummies? And what kind of information can we derive from DNA that's extracted from the mummies? This is an actual photograph of a mummy unwrapped. This is from a museum exhibit. So this is not as it was found. They certainly don't exist like this anymore in Luxor. However, this is it's kind of a diorama at a museum exhibit, although the mummy itself is an actual mummy. It's just been placed and arranged to appear as if it were still in the tomb or preparing to be enclosed in the sarcophagus. So how do you get DNA from something like this? And we've got a big problem because obviously there's a tremendous amount of degradation, the degradation by the chemicals and the agents used to mummify the pharaohs, sheer age and breakdown of the tissue. Obviously, we have biological elements. What if there's certainly bacteria, yeast, molds, not to mention insects, etc., that can come in and eat parts of the decaying body, basically destroying some of the DNA. So we've got a big problem if we want to extract DNA. Where are we going to get it? And of course, what about contamination? So we've got a lot of people involved in the mummification process. There are physicians, religious members who are involved in actually mummifying the body. And so, how do we determine whether the DNA is from the actual individual being that has been mummified, or is it from all these other people? So, if you were to try to extract DNA from a mummy, a carcass that's thousands of years old, where would you look? What do you think?
Inside the bones.
There you go. Great idea. Inside the bones. And in particular, and we've got huge advantages because obviously bones have a medullary cavity, which are filled with cells. Too often, if you have not had either anatomy or physiology at some point in your academic career, know that bone is not dead tissue. In fact, it is very, very living. It's loaded with living cells, and the medullary cavity even has primordial germ cells. It turns out that the bones themselves turn not to be such a great resource of intact DNA, but there is another portion of something that could be considered to be bones where they found the best DNA. And in fact, it is the most intact DNA that's used by forensic scientists today. Anybody want to take a stab at where it might be?
Is it the teeth?
Yes, nice. Yes, it's in the pulp of the tooth. And for some reason, and for, I don't know, probably a million reasons, they can drill into the tooth pulp, extract what is now dried out tissue, and extract a remarkable amount of intact DNA. So really kind of cool. The other side of this, and you've probably, I don't know if you watch any of those crazy TV shows, NCSI, I don't even know them all. All of this where they talk about how they should handle DNA. And it turns out one of the key things is contamination is a huge problem. I'm actually watching, anybody seen the show Clarice on CBS? It's about the FBI agent who was in Silence of the Lambs. Okay, go back, watch Silence of the Lambs. And you could check out Silence of the Lambs, and then this is supposed to be the same FBI agent, but her early career in the FBI. The thing that I thought was fun in the most recent episodes, they talked about isolating mitochondrial DNA from the corpse of an individual that was stored inside a wall for a long period of time, and there was no intact DNA, but they were able to obtain mitochondrial DNA from them. So, and it turned out, I wouldn't have used the words they chose, but they were remarkably correct from a biochemistry point of view. And one of the things that they note is they need to have reference samples of anybody who may have touched the person while they were still alive so that their DNA isn't contaminated. And of course, anybody who does forensics work, as discussed in a couple of our forensics videos, as well as anyone who's working in the area of medical genetics or archaeological genetics, knows that you have to have your DNA profiles online, available in databases so that you know that the DNA you actually want is not because somebody accidentally coughed in a tube. Okay. So we have a situation where they were able, and actually they're more than this, but I've developed uh, a subset of families for us to take a peek at. And they have 7 mummies that were identified: Amenhotep, Tia, Akhenaten, Thuya. Uh, the one in the lower left-hand corner is KV35YL. I affectionately call her KV. KV was in one of the tombs that was looted, and there was no identifying information about her. And in fact, we still don't have a name. We have no historical artifacts as to her name. We just know that she's KV. Tutankhamun, obviously, and Yuya was another that was found. And you can pretty much tell by looking at the sarcophagi whether they were male or female. And so, what would you do? There's some now, again, these are a little bit dated. So, maybe today you might use some of the personal genomics approaches in 23andMe or in ancestry.com. But since that really was not developed at the time this work was done, what other form of DNA analysis could you use to identify these people? Anybody remember? Well, prior to being able to use the single nucleotide polymorphism chips, we had the short tandem repeats. And so we could use the CODIS system that's used to identify criminals in the FBI database to actually identify the mummies and to obtain a DNA profile from the mummies. And in fact, that's what happened. And here is what it looks like. What I've done is I've put together 9 of the 13 short tandem repeats that are done in the CODIS analysis. The first column you can see are squares and circles, and they conform to our, way back when at the beginning of the semester, they conform to our pedigree symbology for males and females, right? Males are squares, females are circles. And so we've got Tutankhamun, Yuya, Amenhotep, and Akhenaten are all males, whereas Thuya, Tia, and of course our little KV are females. So we've got males and females. Notice I've color coded some of these just to make them easy to look at. I don't know, some people don't find it easy to look at. And I do apologize for some of you who may have difficulty distinguishing some of the colors. That's really my fault in choosing the colors. I'm going to have to redo these this summer so that I can recolor everything. But the bottom line is the numerical values. Now, remember, each of the columns, and you can see the label of the columns atop the D13S317, is that particular short tandem repeat, okay? These are the results of looking at the short tandem repeats. And you could see in that first column for the D13 PCR probe, you could see that King Tut has a 10 mer, or 10 copies of the short tandem repeat. And also his other allele is 12 copies. So he's got a 10 and a 12. Think about it in context as that would be cap A and small a. Notice Yuya has 11 repeat, an 11 mer, and a 13 mer. Thuya has a 9 mer and a 12 mer. Tia 11 and 12, and so on throughout the list. And I've done the same thing for each of the other 6 CODIS gene combinations. All right. So each of these combinations can be looked at as a genotype, if you recall the lecture on the CODIS database. Each of these can be considered to be a genotype. Each individual number within a CODIS type is an allele. So these are clearly multi-allelic. And of course, if you combine all of them together, you have a profile that is extremely unique. Now, one thing, just looking at the colors within a column, is notice how many of them are identical. A fairly substantial number of them. And again, just looking at that D13 first column, out of 7 individuals, we've got 1, 2, 3, 4 number 10 alleles and 2 number 11 alleles, and 1, 2, 3, 4, 5 number 12 alleles. So this suggests that we have a very, very high proportion of the same alleles within a very small population. What do you think that tells you about the population? What do you think?
That they're either related or inbreeding?
Yeah, absolutely. Absolutely. That they're certainly related. We know they're all part of the Pharaoh group, but because of the constant brother-sister marriages and consanguineous marriages that aren't brother and sister, there's probably a high degree of inbreeding. Okay. And pretty easy to tease that out. I mean, think about it in the context of your family. You guys drew pedigrees, I assume, for laboratory. Okay. How many genes do you share with your mom or dad? Let me rephrase.
Half from each.
There you go. You get half of each. So I should have said what proportion, not how many. But you share basically half of the genetic information from your mom and dad. So if you're looking within your family, the further you get away from either full brother, full sister, or mom or dad, or parent and offspring, the further away you get from half and half. And yet here we've got a substantially greater than 50% numerical value for a handful of alleles. So this is really a clear indication that not only are they related, but there are probably a lot of similar relationships, meaning a lot of inbreeding throughout the course of the history of the family. So what then can we do with this information? And of course, what I'm going to ask you guys to do, and you can either take a screenshot of this list. I will put, as soon as the video is compiled, I will put it online. What I'm going to ask you to do at the end, and we're going to talk about how to do this in just a second, is to put together a pedigree of King Tut's family from this information. Now, I know somebody told me a year ago, you can now find online, you can find these pedigrees. I'm not asking you to turn them in. This is not a homework assignment. This is something for you to do as an exercise and to give you value in terms of why should I do the exercise if I'll never have to do this again? And the answer is you will have to do this again, because I will have a similar question on the final exam related to taking molecular information and putting together a pedigree. So if you do this ahead of time, you will answer the question very quickly and not have a problem. If you decide you don't want to do this ahead of time, then it may take you a little bit longer. So that's entirely up to you. So take a screenshot if you like. If not, this particular slide will be associated with the video on Blackboard as soon as it's compiled. So probably within about 40 minutes after we finish our session this morning. So I'll get it up pretty quickly. So how do you do this? How do you proceed? Well, the easiest way is to take advantage of the idea that was just mentioned, and that is that an individual gets half the genes from mom, half the genes from dad. So we know if we look at this D13, if we look at the D13, excuse me. All right, sorry, talking too much. Better. If we look at the D13 locus, we see that Tutankhamun is a 10 and a 12. So what does that tell us about his mom and dad? Well, one or the other of the 10 and 11 or 10 and 12 alleles has to be carried by his mom and dad. One will have 10, one will have 12. So let's take a look down. We've got the first of our males, so let's look down. We've got Yuya. And if you look across, Yuya has 11 and 13. We know Yuya is a male. So can Yuya be Tutankhamun's dad?
No.
No, right? Doesn't have 10 or 12. We now have a couple of females. Can Thuya then be Tutankhamun's mom?
Could be.
Maybe, right? She's a possibility. How about Tia?
Also possible.
Right? Maybe. That's okay. Now we've got Amenhotep. Could Amenhotep be his dad?
If possible.
Possible. How about Akhenaten?
Possible.
Possible again. So we can eliminate Yuya as a dad, okay? So we know that Yuya is not the dad, and we've got a few possibles. Let's just stick on the dad side for now. We've got a possibles for dad. So what do? We don't have a definitive. So we go to the next locus. So now let's look at the column for D7. So D7 can have a 10 or a 15. So let's go down. Is Yuya, well, we already know Yuya is not the dad, so it doesn't matter what Yuya have because we know that can't be the dad, okay? So now let's go to Amenhotep. Can Amenhotep be the dad?
Possible.
Possible. How about Akhenaten?
Also possible.
Also possible. So this particular locus is useless for us. right? We couldn't eliminate another one of the other guys. So let's go to the next one. We'll go to the D2 S1338 locus, 16 and 26. And I'm going to drop straight down to Amenhotep. So can Amenhotep be? Well, there's 16, right? So possible. How about Akhenaten? Another 16, damn inbreeding. Maybe they're the same. So let's go to the next D2. This is the D21 S11. So we've got to have a 29 and a 34. So let's drop down again to Amenhotep. 25, 34. Akhenaten, another 34. Okay, so let's go to the next one. So let's go to D16. We've got an 8 and a 13. Let's drop down to Amenhotep. Oh boy, he's got both, so he's possibility. Akhenaten, another 13. Starting to wonder if they're all related. Let's go to D18. D18. Well, Tutankhamun is homozygous for 19. So this is like being Kappa, okay? It's a 1919. So now let's go down and look. How about Amenhotep?
No.
No. How about Akhenaten?
Mm-hmm.
Yep, absolutely. So we got to D18 and we can see on the basis of this information, we can see very clearly that Akhenaten, and by the way, just to make sure, you can go through the other ones if you like, okay? But clearly, Akhenaten, on the basis of all the loci that we've evaluated for now, Akhenaten is Tutankhamun's dad. So now you can start building your pedigree. All right. Now we go through the same process with the females on the list. Looking at D13, we've got 10 and 12. Thuya, if you recall, Tia, and KV. Everybody's a possibility. So we go to the next one. D7, okay, 10 and a 15. Thuya, possible, right? Everybody following? Tia, possible. KV, possible. Okay, let's go to D2, 16 and 26. Oh boy. Thuya, possible. Tia, possible. KV, possible. Okay, let's go to D2, anyone? 29 and 34. Thuya? No. Tia? Possible. KV? Possible. So we've eliminated Thuya, so we're down to Tia or KV. So let's go to the next one, D16, 8 and 13. Guess what? Tia cannot be. His mom, I was rooting for KV all the way. If we can't find your name, you could at least be King Tut's mom. So we've gotten at least to the mom and dad of King Tut. And what I would ask you to do is to spend a little bit of time developing the pedigree from this information. I'm going ahead and stop the share here. Here we go. So I think you can see if you're a little bit of a math geek, if you like puzzles, genetics is right up your line. This is something that's really something geneticists do quite a bit. I find it to be a lot of fun, but then again, I do the New York Times crossword puzzle every morning. So, you just, I enjoy these kinds of mental gymnastics, if you will. They really help to keep your mind fresh and flexible. And I think that's a positive element, especially in higher education. So as soon as we're done, I will post this. I will also post, I don't know. I'll post the answer, but I will trust it to you to only look at it. Only look at it to check your answer. But I certainly would like you to go ahead and give it a good, what do they say? The good old college try. Give it a shot. Okay. It's certainly doable. It's not difficult. I think it's much more fun to do it in person if we were in class together. So it's not quite as much fun, you guys doing it on your own, but feel free to get together with somebody else if you want to go back and forth or to do however you want to get to the answer. We are crashing in as usual to the end of the semester. And as usual, I always want to do more than we have possibly the amount of time to fit in. One thing I do want to mention to you is that the final exam, I think our final exam is supposed to be Monday. Does anybody recall offhand? Not obviously not like 3 days from now, Monday. Anybody remember?
I thought it was May 5th.
Oh, is that what it is?
I don't know. I thought I read May 7th on the thing.
Is that like, wait a minute, I don't remember. Sorry, I meant to look it up. Oh, it could be on Friday, like the last day of finals. It could be. Wait a minute, let me check real quick. 2 seconds.
Yeah, I have that written on my calendar.
Oh, okay. So I guess the question is, what would you like to do? Do you want it? Do you want me to make it available all week? I mean, it's online, so it doesn't matter. It's not like you have to wait around to go home.
Yeah, that sounds good.
Is that okay?
Yeah,
Yeah, I like that idea.
It open for a week.
Yeah.
All right. So how about we tidy up next Friday? We will have we'll go ahead and have our meeting, our regular Friday meeting. It'll be our last one, and then I will make the yeah, and then I'll go ahead and I'll just make it available like Monday through Friday, and you take it when you're able.
Okay.
I think that makes sense. That's okay with me. That's okay with me. There will be one of these problems, very similar, not quite as complicated, but very similar in concept on the exam. I will tell you, I think I told you on the very first day of class, I tell you again, that the first question on my final exam has been the same since 1987. So I ask the same question all the time. One of the fundamentals of genetics that we talk about and continue to talk about every week is how the definition of a gene has changed over time. And the first question on my final exam has always been, for this class anyway, has always been, give me several definitions of a gene. Piece of cake, right? Piece of cake. What do I mean by several definitions of a gene?
Historical definitions and modern definitions.
There you go. Right? Mendel had a definition. He didn't even have the word gene. But we talk about Mendelian genes. Thomas Hunt Morgan had a definition. Watson and Crick had a definition. Bioinformatically, there's a definition, right? In between Watson and Crick and bioinformatics, we know that there's a difference between prokaryotic genes and eukaryotic genes, right? And of course, the last thing we will do next Friday is I will give you the modern, contemporary definition of a gene. How do we think about genes today? Because remember, last time we talked about online Mendelian inheritance in man, 2/3 of what are considered genes, and what by in this context, what I mean is 2/3 of inherited phenotypes, whether diseases or just phenotypes, 2/3 do not conform to the Watson and Crick central dogma definition of a gene. So it means that there's something bigger out there. There is a more complex, more complete definition of a gene in today's world. And we will tidy up the course with that definition. So that always ends up being the first question on the exam, and it really kind of covers the entire span of the course. It covers the entire span of the course. So fun and games. Other than that, I have not written the final yet. It will probably encompass a bunch of questions from the first exam, a bunch of questions from the second exam, and then, of course, things that we've talked about since then, including the question that I've just told you about, my definitions of a gene, and a couple of these, a couple of questions looking at the integration across traditional and more molecular genetic aspects, but always focused on the ideas of inheritance, because that's what this is. This is not a molecular biology course. We're talking about inheritance. So that's what I got. I hope you have fun with the little problem. I still enjoy it, even though I kind of know the answer already. Sorry. I always root for KV. I need shirts, Team KV. All right. Well, anybody got anything? Need something from me?
I just wanted to add a comment, or really more to the conversation a bit about.
Sure, Ash.
So I was just wondering, I'm not sure if I'm confusing the documentary with King Tut's own documentary. I think I'm mixing two of them up. But didn't King Tut and one of his sisters die of the same thing, or it's presumed that they died of the same thing, of a famine or something like that?
Yeah, my understanding was that they both died, and they thought, I think, wasn't it like a Nova documentary, a PBS thing?
I think it was a PBS one.
Yeah, that sounds familiar to me. And I think there were some thoughts that they died of the same disease. I don't recall the famine, but I am not a professor of Egyptian history.
Right. Yeah, because there's this other documentary that came out in regards to a new discovery, and how they found a tomb with a lot of mummified cats, and how it's the first of its
Oh,
kind.
wow. I can think of no better place for cats. No, sorry, bad joke. No, I'll have to look into, or if you find something, just shoot it to me.
Yeah, I could actually send you the name of the documentary.
Oh, that'd be awesome.
You.
That'd be awesome. I'm always interested. I had I actually, just kind of tidying up the story, I had a wonderful visit in my lab with I had a professor after I had been to Egypt a few years later. A professor from Egypt came and spent 6 months in my lab, and it just a wonderful people. I mean, I know sometimes you get a bad taste in your mouth from the news and from hearing about different cultures and different people, but I have nothing but wonderful things to say about my time there. They were the people were wonderful. The food is remarkable. I mean, if you like international food and you're not stuck eating I don't know, whatever the hell we eat in America, like McDonald's cheeseburgers, and you think Taco Bell is Mexican food, then you might be disappointed. But it's just a wonderful culture, wonderful people. I just had a great time. It was really remarkable experience for me. And then having him visit us was even better, because I learned so much more by working with him day to day in the lab. So really kind of cool stuff. All right. Well, I'll look forward to that. You guys take care of yourselves. Enjoy the weekend. Looks like rain on Sunday, so get to the beach tomorrow. Take care, everybody.
Have a great weekend.
You too.
Okay.